everyone, welcome to my YouTube channel. This is Sir Steven, and for this session, we will be discussing genetic disorders as a part of your medical histology laboratory course. Genetic disorders are far more common than is widely appreciated. The lifetime frequency of genetic diseases is estimated to be 670 per 1,000. Furthermore, the genetic diseases encountered in medical practice represent only the tip of the iceberg. That is, those with less extreme genotypic errors that permit full embryonic development and live birth. It is estimated that around 50% of spontaneous abortions during the early months of gestation have a demonstrable chromosomal abnormality, and there are, in addition, numerous smaller detectable errors and many other genetic lesions that are only now being recognized thanks to the advances in DNA sequencing, and about 1% of all the newborn infants possess a gross chromosomal abnormality. And all of these rare diseases with a significant genetic component develops in approximately 5% of individuals younger than age 25 years. How many more mutations remain hidden? So for this session, let's take a look at this diagram which features the examples of a disease located on each of the chromosome. Let's start with the chromosome 1 until the 22nd, or the autosomes, and we have your X and your Y chromosome. So some of which are the DNA test currently available. Let's say, for example, your adrenoleukodystrophy, your azoospermia, Gaucher disease, Ehlers-Danlos syndrome, retinitis pigmentosa, Huntington disease, familial adenomatous polyposis, hemochromatosis, spinocerebellar ataxia, cystic fibrosis, Werner syndrome, melanoma, multiple endocrine neoplasia type 2, sickle cell anemia, phenylketonuria, retinoblastoma, Alzheimer's disease, Tay-Sachs disease, polycystic kidney disease, breast cancer, neurofibromatosis, amyloidosis, myotonic dystrophy, familial hypercholesterolemia, ADA immune deficiency, amyotrophic lateral sclerosis, glucose and galactose, hemophilia, and also your muscular dystrophy. This is a schematic and relatively language neutral cardiagram of a human. Even at low magnification, it gives an overview of the human genome with numbered chromosome pairs and its main changes during the cell cycle. We can see it on the left topmost portion, and also the mitochondrial genome to scale at your bottom right. High magnification or the full size version are clearly depicting a scale to be showing you the chromosome pairs that showing length in terms of million base pairs, and the scale to the right of each chromosome pair showing the designation of the bands and your subbands. It also shows darker and brighter regions as seen on this G banding. Each row is vertically aligned at the centromere level. Your chromosomes are organized into the groups of A to G in regard to sizes, position of the centromere, and sometimes the presence of a chromosomal satellites. The chromosome copy numbers are those seen in G0 and G1 of the cell cycle, and at top center it also shows the state of the chromosome 3 pair in the metaphase of the cell cycle after the synthesis of each of the chromosome. The human mitochondrial genome is to scale on your bottom right. So for the contents of these genetic disorders discussion, we have your chromosomal disorders, the modes of inheritance of monogenic disorders, the Mendelian disorders, the polygenic and the multifactorial disorders, and the disorders of sexual differentiation. For chromosomal disorders, it is defined as the changes in the chromosome number or structure. The normal cells are diploid or 2N, containing 23 pairs of chromosomes or 46 in total. This is 22 pairs again of autosomes and a pair of sex chromosomes, XX in females or XY in males. And these are the classifications of the chromosomal disorders. We have your aneuploidy, for instance, the non-disjunction in your anaphase lag. We have your polyploidy, which will lead to spontaneous abortion, deletion, inversion, and translocation. And later on, we will uh, dig deeper to some of the figures and diagrams that will help us understand what are these chromosomal disorders. For the sex chromosomes, we have your X inactivation and bar body formation. And in extreme karyotype deviations in the sex chromosomes are compatible with life. This is believed to be due to X inactivation, or we call it lionization, and the relatively scanty genetic information carried out by the Y chromosome. We have your bar bodies, also known as your sex chromatin, and your X inactivation. We have also the abnormalities of autosomal chromosomes. Down syndrome, or the trisomy 21, is the most frequent one. The causes is trisomy of the 21st chromosome and translocation. Characteristics are such as like almond-shaped eyes, mental retardation, and many other more. Complications are like congenital heart disease, acute leukemia, and etc. For mothers who would like to screen for Down syndrome, we have your alpha fetoprotein, which is low, human chorionic gonadotrophin, which is high, unconjugated estriol and low, and also lastly, the foregoing assays are referred to as the triple screen. When the assay for elevated serum inhibin A is also performed, it is referred to as your quadruple screen. So mothers can do your triple or quadruple screening. In addition to your Down syndrome, you have your Cradle-Shaw syndrome, the DiGeorge velocardiofacial syndrome, which is the microdeletion of the 22q11, the TCH22 chromosome. We have the Edwards syndrome for trisomy 18 and the Patau syndrome, or the trisomy 13. For the sex chromosomes, we have your Klinefelter syndrome, your XYY syndrome, your Turner syndrome, your XXX syndrome, which is the 47th triple X, and other multi-X chromosome anomalies. With abnormalities due to increased numbers of trinucleotide repeats. For several disorders, it has been found to be associated with the expansion of the number of tandem trinucleotide repeats in certain critical genes. Later on, I'll show you what are these genes and their associated diseases. We have also the Fragile X syndrome. For disorders associated with genomic imprinting, in these hereditary disorders, different phenotypes occur depending on whether an abnormal gene is of maternal or paternal in origin. Imprinting involves transcriptional silencing of the paternal or maternal copies of certain genes during gametogenesis, and for such genes, only one functional copy exists in the individual. Loss of the functional or not imprinted allele by deletion gives rise to your diseases. So this phenomenon is illustrated by two rare syndromes in which the same cytogenetic deletion, the deletion of 15th chromosome, particularly your Q11 and Q13, resulting in different phenotypes in progeny depending on whether the deletions were transmitted by the mother or the father. So we have your paternal transmission, your Prader-Willi syndrome, and for maternal transmission, the Angelman syndrome. Later on, I will show you diagrams that will help us understand what do your genomic imprinting means and the disorders associated with it. Our next classification of the genetic disorders are the modes of inheritance of your monogenic disorders. So we have your autosomal dominant inheritance, autosomal recessive inheritance, X-linked recessive inheritance, other modes of inheritance including your X-linked dominant, which is very rare inheritance, and lastly, the maternal or the mitochondrial inheritance. This is a diagram that will help you understand the pedigree analysis of the different modes of transmission. If for monogenic disorders, we have your autosomal dominant, autosomal recessive, and below the X-linked recessive. And take note of the legend for boxes that has half shade. We have heterozygous male. For circles with half shade, the homozygous female. And for arrows that are pointing on the circles or the squares, these are phenotypically abnormal or those with affected diseases. And the squares are for normal male, and unshaded circles are for normal female. Now for Mendelian disorders. It follows Mendelian principles, and these were classified into different subclassification. We have autosomal dominant disorders such as your adult polycystic kidney disease, your familial hypercholesterolemia, your hereditary hemorrhagic telangiectasia, your hereditary spherocytosis, your Marfan syndrome, neurofibromatosis type 1 and type 2, tuberosclerosis, and your von Hippel-Lindau disease. For autosomal recessive disorders, I will show you a table later on. And we have your lysosomal storage diseases, your glycogen storage diseases, the disorders of the carbohydrate metabolism, such as your galactosemia, disorders of amino acid metabolism, and your cystic fibrosis, such as mucoviscidosis, fibrocystic disease of your pancreas. And we also have your X-linked recessive disorders, such as your Hunter syndrome, your Fabry disease, also known as your angiokeratoma corporis diffusum universale, your classical hemophilia, and your Lish-Nyhan syndrome. Now is your balanced polymorphism. For balanced polymorphism, it refers to the increased incidence of deleterious, usually in homozygotes, alleles among certain populations in environments in which the same allele is being associated with a potential survival advantage, usually in heterozygotes. This condition has been observed in both autosomal and X-linked disorders. We have examples such as your hemoglobin S, your glucose 6 phosphate dehydrogenase deficiency, phenylketonuria, Tay Sachs disease, and your cystic fibrosis. For your polygenic and multifactorial disorders, these are disorders that are more common than monogenic disorders, and they are complex that are being regulated by the protein products of two or more genes. We can also say that environmental factors also play an important role in the modulation of the genetic defects. Common polygenic disorders include ischemic heart disease, diabetes mellitus, hypertension, gout, schizophrenia, bipolar disorders, and neural tube defects. For the disorders of sexual differentiation, these occur when your genetic sex, your gonadal sex, or your genital sex of an individual is discordant. By definition, the genetic sex is determined by the presence or the absence of a Y chromosome, and at least one Y chromosome is necessary for a male gender to manifest. Gonadal sex is determined by the presence of ovaries or testes. The gene responsible for the development of the testis or the sex-determining region of the Y gene is also known as your SRY gene that is being localized to the Y chromosome. For genital sex, which is based on the appearance of your external genitalia, penis or the vagina. For your true hermaphrodite, this rare condition is characterized by both ovarian and testicular tissue, with ambiguous external genitalia and both X and Y chromosomes. On your right is a photograph from 1860 by Nadar of a person displaying ambiguous genitalia, one of a 9-part series. The series may be the earliest medical photographic documentation of an intersex person. One possible mechanism is the parthenogenetic division of a haploid ova into two haploid ova followed by double fertilization and then fusion of the two zygotes in your early embryonic development. We have also a term called pseudohermaphrodite. This organism has the gonads of only one sex, but the appearance of the external genitalia does not correspond to the gonads present. For our male hermaphrodite showing you on your right. For your female hermaphrodite with normal uterus, as what you see it on your right, is a sonographic view and a close-up view of the genitalia. Now let's move on with the review about mutations. So this is an example of a nonsense mutation. This leads to a premature chain termination. Your partial mRNA sequence of your beta globin chain of the hemoglobin showing codons for amino acid 38, 39 to 40, and you will see a point mutation here. It's highlighted in blue green text, and this is the cytosine to uracil in codon 39, which changes a glutamine codon to a stop codon, and hence the protein synthesis stops at your amino acid 38. Next is a 3 base deletion in the common cystic fibrosis allele, which will result in the synthesis of a protein that lacks the amino acid phenylalanine or the 508. Because the deletion is a multiple of 3, this is not a frame shift mutation. This is a point mutation. Single base deletion at your ABO, or also known as your glycosyl transferase locus, leading to a frame shift mutation for the O allele, and this is showing you on below the ABO O allele that has an altered reading frame. This is a 4 base insertion. You will see the nucleotides, the 4 nucleotides, your T, A, T, and C in the hexosaminidase A gene, leading to a frame shift mutation. This mutation is the major cause of Tay-Sachs disease in Ashkenazi Jews. For autosomal dominant disorders, again, they are characterized by the expression in heterozygous state, and they affect males and females equally. Both of the sexes can transmit the disorders. So for autosomal dominant disorders, enzyme proteins are not affected. Instead, the receptors and structural proteins are involved. We have the nervous system, Huntington disease, neurofibromatosis, myotonic dystrophy, and tuberous sclerosis. For urinary, we have polycystic kidney disease. For gastrointestinal, we have your familial polyposis coli. For hematopoietic, you have hereditary spherocytosis or the von Willebrand disease. For skeletal, the Marfan syndrome, the Ehlers-Danlos syndrome, the osteogenesis imperfecta, achondroplasia. For metabolic, we have your familial hypercholesterolemia and your acute intermittent porphyria. For autosomal recessive diseases, it happens when both copies of a gene are mutated. The enzyme proteins are frequently involved. Males and females are affected equally. For the metabolic system, we have cystic fibrosis, phenylketonuria, galactosemia, homocystinuria, lysosomal storage diseases, your alpha-1 antitrypsin deficiency, Wilson disease, hemochromatosis, and your glycogen storage diseases. For hematopoietic, we have your sickle cell anemia with thalassemias. For endocrine, we have the congenital adrenal hyperplasia. For skeletal, Ehlers-Danlos syndrome and alkaptonuria. For nervous, we have your neurogenic muscular atrophies, the Friedreich ataxia, and your spinal muscular atrophy. X-linked disorders are transmitted by heterozygous females to their sons, who will manifest the disease. Female carriers are usually protected because of random inactivation of only one X chromosome. For the musculoskeletal, we have your Duchenne muscular dystrophy. For blood, hemophilia A and B, chronic granulomatous disease, glucose 6-phosphate dehydrogenase deficiency. For immune, we have your agamaglobulinemia and the Viscott-Aldrich syndrome. For the metabolic, you have your diabetes insipidus and Lesch-Nyhan syndrome. And for nervous, the Fragile X syndrome. This is a figure of a possible metabolic pathway in which a substrate is converted to an end product by a series of enzymatic reactions. M1 and M2 are the products of your minor pathway. We have here a big table showing you the biochemical and a molecular basis of some Mendelian disorders, and they are classified according to their protein type or function, such as your enzyme inhibitors, your receptors, your transport oxygen or in ion channels, and for structural, we have your extracellular or cell membrane, hemostasis, and lastly, the growth regulation. This is a figure of your low density lipoprotein metabolism, or the LDL, and the role of the liver in its synthesis and clearance. Your lipolysis of your very low density lipoprotein, or your VLDL, by the lipoprotein lipase in the capillaries releases triglycerides. These triglycerides are then stored in fat cells and used as a source of energy in your skeletal muscles. Your APOC, or apolipoprotein C, your ApoE, your apolipoprotein E, your B100, your apolipoprotein B100, your ApoB, IDL stands for your intermediate density lipoprotein. This is a low density lipoprotein LDL receptor pathway. You may pause on this particular slide to let understand the regulation of cholesterol metabolism. We have your ApoB100, your apolipoprotein B100, or your ApoB, your MHG, CoA, and 3-hydroxy-3-methylglutaryl coenzyme A. This is the classification of your low-density lipoprotein receptor mutations based on abnormal function of the mutant protein. These mutations will disrupt the receptor synthesis in the endoplasmic reticulum transport to the Golgi complex, the also binding of apoprotein ligands and the clustering in coated pits and recycling in endosomes. Not shown here is the class 6 mutation, in which initial targeting of the receptor to the basolateral membrane fails to happen. Each class is heterogeneous at the DNA level, and these are modified. The LDL receptor locus shown here is common in familial hypercholesterolemia. This is another figure for the synthesis and the intracellular transport of your lysosomal enzymes. And to accompany this figure, lysosomal storage diseases and its pathogenesis is being shown in this figure. In this example, a complex substrate is normally degraded by a series of lysosomal enzymes such as your A, your B, and your C into your soluble end products. And if there is a deficiency or the malfunction of one of the enzymes, for example, B, the catabolism is incomplete and insoluble intermediates will accumulate in your lysosomes. In addition to this primary storage, secondary storage and toxic effects result from defective autophagy. And to add, these are the common lysosomal storage diseases: your glycogenosis, sphingolipidosis, sulfatidosis, mucopolysaccharidosis, mucolipidosis, other diseases such as for complex carbohydrates such as your fucosidosis, mannosidosis, aspartyl glycosaminuria, and other lysosomal storage diseases such as your Wolman disease. This is the ganglion cells in your Tay-Sachs disease. Under the light microscope in letter A, this has a large neuron. You will see very obvious lipid vacuolation. In letter B, a portion of a neuron under the electron microscope shows prominent lysosomes with whorled configurations. A part of the nucleus is shown above. Next disease is Niemann-Pick disease in liver. The hepatocytes and your Kupffer cells have a foamy, vacuolated appearance due to the deposition of lipids, as shown here in this micrograph. Next is Gaucher disease involving the bone marrow. In these two micrographs, the Gaucher cells are plump macrophages that characteristically have the appearance in the cytoplasm of crumpled tissue paper due to the accumulation of your glucocerebrosidase. This letter A has the using the right stain. Letter B is using hematoxylin and eosin stains. This is a figure just to give you a review about biochemistry, the pathways of glycogen metabolisms, and we have the asterisks. These asterisks mark the enzyme deficiencies associated with glycogen storage diseases. Roman numerals indicate the type of glycogen storage disease associated with a given enzyme deficiency. We have types 5 and 6 that result from deficiencies of muscle and liver phosphorylases, respectively. These are the principal subgroups of glycogenosis. We have the clinico-pathologic categories, hepatic type, myopathic type, and miscellaneous types. Specifically, we have your hepatorenal von Gierke disease for type 1, myocardial disease for type 5, and we have the generalized glycogenosis or the Pompe disease for type 2. Their morphologic changes, enzyme deficiency, and the clinical features are shown in this figure. They have a normal glycogen metabolism in the liver and the skeletal muscles. In letter B, we have the effects of an inherited deficiency of hepatic enzymes involved in glycogen metabolism. Letter C, you will observe the consequences of a genetic deficiency in the enzymes that metabolize glycogen in skeletal muscles, such as this. Next figure is the G-banded karyotype. An example from a normal male, you will see that they have 46 chromosomes and the X and the Y. Also shown is the banding pattern of the X chromosome with nomenclature of arms, the regions, the bands, and the subbands. These are examples or types of chromosomal rearrangements: balanced reciprocal, centric fusion or the Robertsonian, isochromosomes. We have inversions such as paracentric and pericentric. We have deletions that will produce fragments and ring chromosomes. This is the fluorescence in situ hybridization analysis of an interface nucleus using a locus-specific probes to chromosome 13, which is in green, and chromosome 21 in red, revealing the 3 red signals consistent with trisomy 21, also known as Down syndrome. We have the clinical features and karyotypes of selected autosomal trisomies. We have the trisomy 21 Down syndrome, the trisomy 18 Edwards syndrome, which is in incidence 1 in 8,000 births, and we have the trisomy 13 Dipatau syndrome in their incidence in 1 in 15,000 births. You may pause the video and try to check the clinical features and the karyotypes of these trisomies. This is a fluorescence in situ hybridization of both the metaphase chromosomes and an interface cell from a patient with the George syndrome, demonstrating the deletion of a probe that maps to chromosome 22q11.2. The 22q11.2 probe is in red, and the control probe you will see is localized to 22q, which is in green. The metaphase spread shows one chromosome 22 with both a green signal or the control probe and a red signal from the 22q11.2 probe. The other chromosome 22 shows only hybridization with the control probe in green, but no red 22q11.2 signal since there is a deletion of this chromosome. The interface cell also shows a hybridization pattern consistent with the deletion of chromosome 22q11.2. Another example is the clinical features in karyotypes of the Turner syndrome, which has an incidence of 1 in 3,000 female births. This is the examples of your trinucleotide repeat disorders. As we expand your trinucleotide repeats, this is a very important genetic cause of a lot of human diseases, particularly your neurodegenerative disorders. The discovery in 1991 of their expanding their trinucleotide repeats as a cross of your Fragile X syndrome or your FXS was a landmark in human genetics. Since then, the origins of about 40 human diseases and the expansions affecting non-coding regions and expansions affecting coding regions are shown here with their diseases, their gene, their locus, the protein, the repeats, and their number of repeats in normal versus in disease individuals. These are the sites of expansion and the affected sequence in selected diseases caused by nucleotide repeat mutations. UTR stands for untranslated regions. We have the mechanisms: transcriptional silencing, loss of protein function for Fragile X syndrome, transcriptional dysregulation, accumulation of toxic mRNA for Fragile X tremor ataxia, Fragile X associated primary ovarian insufficiency. For transcriptional silencing and loss of protein function, we have the Friedreich ataxia, and for polyglutamine expansions with misfolding, a toxic gain of function in Huntington disease. An illustration by Patricia Howard Pebbles, University of Texas Southwestern Medical Center, Dallas, Texas, showing you a Fragile X as seen as discontinuity of staining. This is a pedigree of the Fragile X. Note that in the first generation, all sons are normal and all females are carriers. During oogenesis in the carrier female, pre-mutation expands to full mutation. In the next generation, you will see all the females who inherit the X with full mutation are affected. However, only 50% of the females who inherit the full mutation are affected, and only mildly. Not shown are the Fragile X associated ataxia or tremor and Fragile X associated primary ovarian failure that can occur in permutation carriers. This is a model for the action of familial mental retardation protein or your FMRP in your neurons. This is modified from new insights into the Fragile X syndrome for molecules to neurobehavior in 2003. Another disease is the pedigree of your Leber hereditary optic neuropathy. This is a disorder caused by a mutation in the mitochondrial DNA. Note that all the progeny of an affected male in shaded squares are normal, but all children, the male and female of the affected female, in shaded circles manifest the disease to a variable degree as discussed in your text. This is the diagrammatic representation of your Prader-Willi and Angelman syndromes. In Prader-Willi syndrome, there is a deletion of your band Q12 on the long arm of the paternal chromosome 15. Genes in this region of the maternal chromosome 15 are imprinted, so there is complete loss of their functions. Patients with intellectual disability, short stature, hypotonia, hyperphagia, small hands and feet, and hypogonadism. In Angelman syndrome, there is a deletion of the same region from the maternal chromosome, and genes on the corresponding region of paternal chromosome 15 are imprinted. These patients have intellectual disability, ataxia, seizures, and inappropriate laughter. This is an example of a single base extension analysis of a polymerase chain reaction product. We use a primer to interrogate a single base position. The nucleotides complementary to the mutant and the wild type bases at the queried position are labeled here using your fluorescently labeled nucleotides or fluorophores, such that incorporation yields fluorescent signals of varying intensity based on the ratio of mutant to wild type DNA being present. This is the diagnostic application of your PCR or the polymerase chain reaction and southern blot analysis in Fragile X syndrome. With PCR, the differences in the size of CGG repeats between normal and premutation give rise to products of different sizes and mobility. With a full mutation, the region between the primers is too large to be amplified by your conventional PCR. In southern blot analysis, the DNA is cut by enzymes that flank the CGG repeat region, and this is then probed with your complementary DNA that is binding to the affected part of the gene. A single small band is seen in normal males, a band of higher molecular weight is seen in males with premutation, and very large, usually diffuse band is seen with males with the full mutation. This is a figure of the analysis of a copy number variation via single nucleotide polymorphism or SNP cytogenomic array. Genomic DNA is labeled and hybridized here to an array containing potentially millions of probe spots. The copy number is determined by overall intensity and their genotype is determined by allelic ratio. The example shown here is the P arm of the chromosome 12 in a patient with pediatric leukemia. The normal areas, such as green, show the neutral diploid DNA content, and the zygosity plot shows the expected ratio of AA, AB, and BB SNP genotypes. The anomalous area in red shows decreased overall intensity, and the zygosity plot shows the absence of the mixed AB genotype, indicating a full heterozygous deletion. This is DNA polymorphism resulting from a variable number of CA repeats. The three alleles A produce polymerase chain reaction products of different sizes. In B, the identifying their origins from specific chromosome in this example depicted allele C is linked to a mutation responsible for autosomal dominant polycystic kidney disease. The application of these to detect progeny carrying the disease-related gene or the red symbols is illustrated in one hypothetical pedigree. The males are in squares, the females are in circles. And lastly, we have to review the principle of next-generation sequencing. There are several alternative approaches that are currently available for the NGS, and one of the most common used platforms is illustrated here. In letter A, short fragments of your genomic DNA template to 100 going to 500 base pairs in length are immobilized on a solid phase platform such as your glass slide using universal capture primers that are complementary to adapters that have previously been added to ends of the template fragments. The addition of fluorescently labeled complementary nucleotides, let's say one pair template DNA per cycle, occurs in a massively parallel fashion at millions of templates immobilized on the solid phase at the same time. A four-color imaging camera captures the fluorescence emanating from each template location, which are corresponding to the specific incorporated nucleotide, following which the fluorescent dye is cleaved and washed away, and the entire circle is being repeated. In letter B, there is a powerful computational programs that can decipher the images to generate sequences complementary to the template DNA at the end of one run. And these sequences are then mapped back to the reference genomic sequence to identify the alterations. And that's all for the genetic disorders. Up next are the diseases of your immune system. Thank you so much for watching, and don't forget to like and subscribe this YouTube channel. Have a nice day, everyone.